trying to get rid of this little. Okay, thank you very much. This is a real pleasure and a real honor to be here this afternoon, this morning. I'm going to confess that my title is somewhat cryptic. What is a supernumerary axon, and why is one axon not good enough? So to answer the second question, I'm going to turn to a remarkable individual by the name of Ramon Cajal. Cajal, based entirely on very careful anatomical observations over 120 years ago, made the following observations and conclusions. He argued that neurons were essentially composed of two compartments. Back here. In blue, the dendrite tree, which is formed by a number of branches which emerge from the cell body, and then they divide to form a tree. In contrast, the second compartment, which I've shown here in orange, the axon, is always composed of only one process emerging from the cell body, right here. It typically has a uniform diameter, and when it gets near termination, it branches at right angles, and there's all sorts of very interesting swellings on the end. Cajal also found that other axons came in and terminated on the dendrite tree. And based on these observations, he coined what he referred to as the law of dynamic polarization. What he meant by this, and I'm going to now use a modern description of what he was thinking about, is that axons release neurotransmitters, and when they do, they generate signals on the dendrite tree. I've indicated these as pluses or minuses, indicating IPSPs or EPSPs. These signals are transferred down through the dendrite tree, and if they reach a critical threshold, an action potential is generated. So, in Cajal's words, the dendrites actually receive information. In contrast, the axon transmits information, and although Cajal didn't know this at the time, we now know this means the axon, the action potential travels down the axon. And then the whole process of releasing a neurotransmitter, binding to receptors and other neurons begins again. Now, from this perspective, one axon is ideal. But what happens after exotomy? The distal part of the axon degenerates. My goal this morning is to persuade you that when this happens, there is a very nasty environment right here which prevents regeneration. And that under these circumstances, new axons appear. We call them supernumerary axons. Supernumerary is an elegant word for extra. What makes this very curious is they emerge from distal dendrites. And if I'm successful, it's not to my credit, it's to the credit of Monica Hoover-Hess, Victoria McDermott, Claire Meehan, Keith Enrich, and of course, thanks to the Barbara Turnbull Foundation and CIHR for funding this work. Now, the story begins with a very simple experiment. I'm actually not going to use a model of spinal cord injury, I'm going to use a model of peripheral nerve injury, in which we simply transect a number of nerves going to dorsal neck muscles, encapsulate them in vortex sleeves. This is designed to prevent reinnervation and mimic the failure of CNS neurons to regenerate and reinnervate. Sew the animal up and wait. Several weeks later, we look at the structure of the cells whose axons have been transected. Now, this is an example of a dendrite tree of a motor neuron whose axon was intact. Now, these are very impressive structures. If you take all these bits and pieces, this, by the way, is just only the dendrites, add them all up, the total length would be 70 to 100 millimeters in length. This is about the width from my finger to my thumb. What happens after exotomy? You see a very different story. This is 10 weeks post-exotomy. You see these very odd arborizations which emerge from distal dendrites. The story takes a really peculiar twist when you stain this tissue for a protein called MAP2AB. MAP2AB is a specific, unique marker for dendrites. And what I'm showing you here in gold or yellow is where we do not find MAP2AB staining. Just to illustrate this, I've highlighted a couple of examples down here. It's very peculiar. The process is emerging from dendrites, which are not dendrites. In other experiments, we look at other neurons. This is another one 10 weeks post-autotomy. You see some peculiar looking processes off the distal dendrites, here, here, and here, a couple up in here. In this case, we've stained the tissue with an antibody to GAP43. Now, GAP43 is a specific marker for growing axons. In this case, the goal indicates the presence of GAP43. So what we appear to have is a harbinger of a supernumerary axon emerging from a distal dendrite. If we wait 35 weeks post-autotomy, this is just the evidence showing that these are GAP43 positive. If we wait 35 weeks, the dendrite trees haven't changed that much more. But what I want to draw your attention to is this process right here. When we traced it back to the cell body, we went all the way back without a single branch. It looks like it comes from this axon, but in fact it went to the cell body. This entire process is mapped to AB negative. This is by the way a horizontal view. We look at this in the transverse view. It's shown over here. So here's the left side of the gray matter and the right side. You can see that this process, the supernumerary axon, travels dorsally, turns right up here, goes immediately across the spinal cord, and terminates in the opposite side. The exotomy has generated a hybrid. This is cells both a motor neuron and a conditional neuron. I want to highlight this zone right here. I blow it up. You can see the number of right-angled branches, which any self-respecting axon should have, and some swellings. Difficult to know exactly what these swellings mean unless we go to the electron microscopic level. And that's what I've done here. This shows electron micrographs of two sections. They're serial, so one after the other. They're very, very thin. And what I want to draw your attention to is this very dense zone right here and here. This is what the stain that we use to visualize these cells looks like under the electron microscope. And there's good news and bad news here. I'm very disappointed that I can't really see the ultrastructure here. But what I can see is what this process is in opposition to. There's a very long process here. We go to the neighboring section that's even longer as we cut farther into it. There's lots of microtubules, a few mitochondria. These processes are dendrites. So we've got evidence from this that these swellings are doing what an axon should do. They're snuggling up to dendrites. Thus far, everything I've talked about is focused on structural changes and the characteristics of the supernumerary axons. I now want to refer to something I'm going to call functional features, although I'm still going to use anatomical techniques. This green object here is the terminal arborization of another one of these supernumerary axons 35 weeks post-autotomy. I want you to focus in this area right here. There's a very nice distinct swelling. I'm going to blow it up and show it again over here. So here's the swelling. This is the same region of the micrograph, only now I'm only going to look at the distribution of a protein called SP2. The SP2 is critical for the release of neurotransmitters. Now, I hope you can see that right at this arrowhead is a little red spot. If we superimpose this image and this one, you see this turn slightly yellow, indicating that these swellings contain the very protein one needs to release neurotransmitters. Now let's look at another side of what an axon should do. Axons should not only release neurotransmitters, they should also initiate and conduct action potentials. So this shows one of the cell bodies of a motor neuron whose axon was cut 35 weeks earlier. There's these monstrous primary dendrites coming off. What I want to draw your attention to is these two very thin processes, right here and right here. We traced these and discovered they turned into supernumerary axons. Now, in this tissue, we've stained using an antibody which recognizes voltage-dependent sodium channels. Voltage-dependent sodium channels are crucial for generating action potentials. So if I blow up these areas over here, this is the one on the right, this is the one on the left here, the neighboring panels show the distribution of the stain after staining the tissue with the pan-NAV antibody. And I hope you can see there's a little red line here corresponding to this region, just where it comes off the cell body. There's a slightly fainter one, but once again, it's exactly like the process comes off the cell body. This is ideally suited to initiate action potentials. In fact, this is what normal cells look like. If we now turn our attention to the rest of the supernumerary axon, the part that's away down here, several hundred, if not 1,000 microns away, and we look at a small zone right here where it's quite twisty and turning. You can see it right here. This is a series of confocal images which have been stacked together. I'm going to extract two of those and show them here. So this little zone is right up there. We've now stained the tissue with an antibody to NAV 1.2. NAV 1.2 is the sodium channel which conducts action potentials in unmyelinated axons. And I hope you can see, if you superimpose these two, that this process is ringed by the NAV 1.2 staining, implying this process on its membrane has NAV 1.2 channels. So this is my final slide, and I'd like to introduce a new term. The term is PDSA, dendrite-derived supernumerary axon. The conditions under which we believe these occur is when the original axon cannot regenerate. And when this happens, dendrites give rise to supernumerary axons. They have the ability, we think, to conduct action potentials thanks to the sodium channels along the branches as they go to termination, and initiate action potentials at the junction between the axon and the soma. In addition, as shown here and here, these terminals contain sp2, a protein critical for synaptic neurotransmitter release, and they make contacts with dendrites. Now I want to stress, much like Rob, the story does not end here, but I think my time